As you already know, biosynthesis of proteins is under direct control of DNA in most cases, or else under the control of genetic RNA, where DNA is absent. Information for structure of a polypeptide is stored in a polynucleotide chain. Sequences of bases in a particular segment of a polypeptide chain will determine the sequence of amino acids in a particular polypeptide. The relationship, popularly known as central dogma, explains how protein synthesis is controlled by nucleic acids. There are two major steps involved in protein synthesis. Number one, transcription, and number two, translation. Transcription involves transfer of genetic information from DNA to mRNA, and translation involves translation of language of nucleic acids into data proteins. Transcription will be discussed in detail in the present topic. Now, what transcription means? Transcription is the synthesis of RNA which carries the genetic information present in DNA. The DNA is double-stranded and can theoretically code for two separate RNA molecules. However, it has been found that only one of the two strands of the gene is transcribed. Only in a few exceptional cases, both strands are transcribed. This is possible because the promoter is asymmetrical and unidirectional. The DNA strand which have a sequence homology with the RNA is known as the coding strand. The second strand, which is complementary to RNA and serves as the template for RNA synthesis, is known as the non-coding strand. It is therefore a misnomenclature, as it is the non-coding strand which is in fact transcribed to form the primary transcript. Here, we will discuss transcription in both prokaryotes as well as in eukaryotes. The principal enzyme involved in transcription is the DNA-dependent RNA polymerase. To understand the transcription, it is necessary to learn more about the RNA polymerase. The bacterial RNA polymerase consists of 5 polypeptide chains, including 2 chains of alpha polypeptide, 1 chain of beta and beta dash, and sigma polypeptide. The RNA polymerase molecule thus can be represented as alpha 2, beta, beta dash, and sigma, in which the attachment of sigma factor is not very firm, so that the coenzyme can be easily isolated. Once RNA synthesis is initiated, sigma dissociates after RNA is 8 to 9 bases long, and then the core enzyme brings about elongation of mRNA. The dissociated sigma factor may again combine with core enzyme to form RNA polymerase holoenzyme. The entire process of transcription can be divided into following steps. Number 1, template recognition. Number 2, initiation. Number 3, elongation. And number 4, termination. Let us discuss them one by one. Number 1 is template recognition. RNA polymerase has to recognize the correct region of the gene. Gets bind at that site, which is -35 region. The size of the RNA polymerase is such that about 60 nucleotides in the gene are involved in binding of the enzyme to the template. Sigma factor has a vital role in specific binding of this enzyme with the template. The binding of core enzyme without sigma factor to DNA is not promoter specific. The promoter enzyme complex is formed only with the help of sigma factor which is very important for the transcription. RNA polymerase sometimes binds at a site other than the promoter site and is known as loose binding. Sigma factor is responsible for the specific binding of RNA polymerase and reduces the chances of loose binding to a greater extent. RNA synthesis by RNA polymerase has following steps. Number 1, the holoenzyme binds at promoter site, forming the closed promoter complex in which DNA remains double helical. Number 2, the closed complex isomerizes and causes unwinding and separation of DNA strands to form open promoter complex. Number 3, after unwinding, only one of the two strands is copied, which is achieved by incorporation of nucleotides initially without movement of enzyme, leading to the formation of RNA chain up to 9 bases in length. During the incorporation of these 9 bases, at every step, there is a possibility for the release of this small RNA chain, a process described as abortive initiation. A cycle of abortive initiation usually occurs, generating a series of short oligonucleotides before initiation is usually successful. Number 4, once initiation succeeds, the sigma factor of RNA polymerase dissociates. Number 5, the dissociation of sigma factor marks the entry of Nus-A protein, which helps elongation and promotes pausing and termination at specific sites. Core enzyme now undergoes a major conformational rearrangement and a stable elongation complex is formed. This complex moves along DNA, synthesizing RNA all along its path at a rate of about 40 bases per second at 37 degrees centigrade. Elongation of RNA transcript continues till an unstable termination complex is formed. The termination of mRNA chain in prokaryotes is brought about by certain termination signals on DNA. These DNA sequences providing termination signals are called terminators. Once the enzyme hits the terminator, it falls off the template and the transcription stops. The termination signal, whenever found on DNA, can be of two types. Number 1, Roho-dependent termination and Roho-independent termination. First, let us discuss Roho-dependent termination and then Roho-independent termination. Roho-dependent termination. The termination factor Roho helps in termination of transcription. Now, what is Roho? Roho is a protein of 46 kD and its active form is a hexamer having a total mass of 275 kD. Roho attaches to the RNA chain and moves along the growing RNA chain and chases RNA polymerase. Once Roho catches the RNA polymerase, it results in chain termination. How the termination takes place is very much important and interesting. When RNA polymerase hits the terminator sequences, it pauses for a short time and during that time Roho reaches to the RNA polymerase and forces it to fall from the template. Once RNA polymerase is detached, the RNA chain also comes off and transcription terminates. Now, Roho-independent termination. Definite region of intrinsic sequences which causes the termination of RNA chain in some genes. This may be due to two guanine cytosine rich stretches at the end of RNA transcript which are complementary to each other. These two guanine cytosine rich stretches forms an intramolecular hairpin structure. This type of structure is thermodynamically unstable and causes the fall of mRNA from the DNA template. When the RNA is detached, the RNA polymerase also falls off from the template and the termination of transcription occurs. This type of termination shows that the structure of RNA itself can cause its own termination from the DNA chain. The eukaryotes have more than one type of RNA polymerase. Based on the activity to alpha-amanitin, an antibiotic which inhibits mRNA synthesis. 3 classes of RNA polymerases have been identified which are involved in transcription of different classes of eukaryotic genes. The size of eukaryotic RNA polymerase are greater than 500 kD in size, and it has 2 large subunits of 200 kD and 140 kD. The 200 kD subunit is similar to beta- subunit of E. coli RNA polymerase and have similar function of template binding. Besides these 2 proteins, it also has 10 different small subunits. A subunit of Pol II, which has similarity with one of the subunits present in Pol I and also in Pol III, is similar to alpha subunit of E. coli enzyme and helps in enzyme assembly. Apart from the RNA polymerase, different transcription factors are also involved in transcription. Promoter sites for initiation of transcription in eukaryotes. Promoters of RNA polymerase first could not be initially studied since all genes for rRNA were similar. Promoters for RNA polymerase third, on the other hand, had some unusual downstream promoters. However, for RNA polymerase second, several hundred eukaryotic genes have now been sequenced and their promoters studied, revealing some general features in 3 regions located at start point, centered at sites lying between -25 to -100 base pair. The least effective of these 3 regions is the Tata or Hogness box, located 20 base pair upstream to the start point. The Tata box is surrounded by guanine cytosine rich sequences and is comparable to Pribno box of prokaryotes. Further, upstream is another sequence called CAT box, which, being necessary for initiation, is conserved in some promoters, but is not necessary in some other genes. The sequence lies between -70 and -80 base pair. Another sequence of G, 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 C, G, G, which is also called GC box, is found in 1 or more copies at -60 or -100 base pair upstream in any orientation in several genes. It has been shown that CAT and GC box determine the efficiency of transcription, while Tata box aligns RNA polymerase at proper site with the help of transcription factor 2D, TF2D, and other transcription factors. In eukaryotes, the initiation is more complex. It involves a number of specific transcription factors. The process has been followed for the polymerase second action, resulting in the synthesis of mRNA. The process is essentially similar to Pol I and Pol III. For initiation, it requires a number of transacting factors along with RNA polymerase. The transacting factors, which are products of various regulatory genes, bind to either DNA or to each other or to RNA polymerase. They can also bind in various combinations. All the transcription factors involved with Pol II are called transcription factor 2. First, the factor transcription 2D binds to Tata box, covering about 25 nucleotides within the -17 to -42 region. Now, transcription factor 2A associates itself to the complex, further extending the protected region towards upstream up to -55 to -80 region. On the other hand, transcription factor 2B associates itself protecting the region at -10 to +10. It binds to two strands in a non-symmetrical manner. This complex prepares the stage for binding of RNA polymerase II, which covers up to +15 region on template strand and 5 extra nucleotides on the non-template strand. Finally, transcription factor 2E joins extending the protection up to +30 region. Once the entire complex has been assembled, the incorporation of first nucleotide takes place. Transcription factors and elongation of RNA chains in eukaryotes. Certain accessory proteins of transcription called the elongation factors enhance the overall activity of RNA polymerase II, leading to increase in elongation rate. At least two such proteins are known. Number one, the transcription factor TF2F accelerates RNA chain growth relatively uniformly in concert with RNA polymerase II. During RNA elongation, transcription factor 2F remains attached to the RNA polymerase, and all of the other transcription factors have dissociated from PIC, pre-initiation complex. Number 2, transcription factor TF2S helps elongation of RNA chain by relieving the obstructions in the path of some elongation. Transcription factor 2S is known to function by first causing hydrolytic cleavage at 3' end of RNA chain, which are stuck and cannot elongate. Thus, RNA polymerase moves backwards under the direction of TF2S before it moves forwards through the block to elongation. In eukaryotes, the actual termination of RNA polymerase II activity during termination may take place through termination sites similar to those found in prokaryotes. But these termination sites are believed to be present away from the site of 3' end of mRNA. Obviously, 3' end of mRNA will be generated due to post-transcriptional cleavage. This cleavage at the end is believed to be achieved by what is popularly called small nuclear RNA protein complex. Small nuclear RNA protein complex used for post-transcription cleavage has not been identified so far, but is believed to be certainly different from the U1 small nuclear RNA protein complex, which is believed to be involved in intron splicing in split genes. Moreover, a sequence 5' AAUAAA has been found just on the 5' side of poly A addition site in several eukaryotic mRNAs. The poly A tail is added to 3' end of eukaryotic mRNA after processing of precursor mRNA. The sequence AAUAAA in mRNA 3' end seems to be common in eukaryotic mRNA, and mutation in this sequence cause elongation of mRNA. This will suggest that this sequence contains the signal for endonucleolytic post-transcriptional cleavage. This sequence therefore is not involved in the termination of the synthesis of mRNA, but helps in generating 3' end later through endonuclease cleavage, in which small nuclear RNA protein complex helps in an unknown manner.